Okay, I'll adjust it. Good morning, everyone. Welcome to AWS 2017. Let me give you a short introduction about AWS. The annual work seminar, in its culminant avatar, germinated in 2012 in one of the student meetings as a felt need for a platform where young researchers and practitioners within A-Tree would get to communicate their work, interact, and share insights and feedback. Since then, AWS has been organized twice in the years 2013 and 15, making this edition the third. At the AWS, A-Tree faculty, students, research associates, and representatives of our community conservation centers discuss research, policy, and outreach activities, results, as well as proposals. Dissemination to and feedback from the entire A-Tree community are the two main objectives of the AWS. May I now request our director, Dr. Karthik Shankar, to give us the inaugural address.
That sounds way too formal for inaugural address and all that. Let's make it opening remarks. So yesterday we were greeting the new batch of A-Tree PhD students, which is always very exciting for us. And the convener, as a sort of parting shot, said, "I hope you guys have heard the 1970s rock hit Hotel California. If you remember the lines, it goes, you can." And he was referring to A-Tree, you can check out any time you want, but you can never leave. And I was like, this is not a good thing to say to PhD students on their first day, right? But I feel a bit like that myself, as you pointed out. I was here as a fellow for a few years, and we organized what was the previous avatar of the AWS. I think it was called the Annual Research and Conservation Seminar, and this was 2006. And in this day and age, I think the appropriate term for that would be a prequel. My strong and fond memories of the research meeting, which actually, and some of the oldies and goldies might be able to tell me that I think that was the first A-Tree-wide seminar that we had organized, and we had it in Royal Orchid, and we had almost everybody who was at A-Tree was much smaller than spoke about something or the other. I remember myself the excitement that the annual seminar brings for us in being able to share work with each other. I wanted to particularly thank the students for organizing this event. I think it's one of the remarkable things about this the generation of students that we've been blessed to have in A-Tree and in our community over the last decade or so, that one is able to leave the organizations of these events entirely in their hands, and they're done exceptionally well. And so I look forward to what one of our plenary speakers has already described as a packed program. I'd like to thank all the plenary speakers who've done us the honor of joining us. All of our advisory board and governing board members who are present with us today. Several more will be joining us during the course of the next 2 or 3 days. And I thank everybody for being here and participating so enthusiastically in advance. So with that, I'll take refuge in my jet lag for not being more eloquent, but you're probably glad that I'm not going on forever and ever. So without further ado, let's get the show on the road. Ramya, back to you.
Well, no conference is complete without a proceedings book. May I request Karthik, Amita, and Siddharth to come up on stage to release the proceedings books for AWS 2017, which also includes abstracts from the previous 2 editions. So, No. So let me bring your attention back to what will be the focal theme of AWS this time around: long-term monitoring research. ATRI has been a part of several long-term monitoring research projects for several years. Faculty members and researchers working on these projects will be invited to share their experiences in planning and executing these LTMR-based projects or long-term monitoring research projects. With that, I will get out of your hair and invite Dr. TG to start the address for today.
Yeah, good morning, everybody. I really feel like a real oldie now. Long-term monitoring has made some of us sweat. I didn't oldie here. I really thank AWS for giving not only me an opportunity, but a lot of people involved with long-term monitoring in ATRI, an opportunity to really showcase some of the work and also share some of the experiences of doing such work across several decades. So I really thank AWS for that. And okay. Okay, so my talk is titled like a peep into the past. That's the title I gave to the AWS, and I've added a little bit of the future. So it's no point talking in the past without talking a little bit about the future. So I'm also very grateful to the colleagues, Siddhappa, Priyadarshan, Ganesan, and Subhadra, without whom much of this work would never have happened, and probably without whom I would have not done this work as well. So I'm really grateful for them as well. Before I actually launch onto this, so my talk is, though it's about long-term monitoring, I realized that while just putting it together, I just have 40 slides. And then I was going through some of the presentations for 15 minutes, there were more than 45 slides. So you probably see me here for only about 15 minutes, so you don't have to worry too much about it. Okay. Okay, fine. Okay, briefly, so this is my outline of my talk. I'll try to define what's an LTM, because that's, I mean, somebody says the lion-tailed macaque, and it's very commonly used for a lion-tailed macaque than for a long-term monitoring. But often when I talk about long-term monitoring, people do ask, what is long-term? So we'll go through a little bit of that definition, and why did we start such a work in ATRI? And what do we monitor? And I'll go a little bit on what we have been doing in KMPR, which is the Kalakkad Munnar Tiger Reserve, where much of the work has happened. And then the learnings from such programs, because that's going to tell you what you want to do in the future. and of course, way ahead. Okay, what is long-term monitoring? There's pretty the whole list of definitions of that, but I just picked up a couple of them. One says that in 1986, Strayer said that continues beyond the generation time of a dominant organism in an ecosystem are sufficiently long to quantify the key processes which structure the ecosystem under investigation. Well, this is basically, you're looking at the generation time of an organism. So if you look at whales or you look at trees, you have long generation times. So you need many decades as well. But if you look at the bacteria, probably you can see the whole thing in a test tube within a month. So long term differs between the taxa, I mean, how long it lives. And also in terms of ecosystem it lives. Whereas a little later in 2010, there's a very empirical kind of definition, like repeated field-based empirical measurements collected continuously and then analyzed for at least 10 years. Okay, so, and then came in roughly around the time of 2010, anthropogenic effects. I mean, we talk about anthropogenic effects, several forms of anthropogenic, into the long-term monitoring concept. And it also scaled up the time frame from a few years to several decades, centuries, and beyond that. And also include several interesting stuff like global warming, sunspot cycles, and stuff like into the monitoring schedules. Well, that sort of got termed as long-term monitoring. Okay, what do we do in such monitoring? These are based on National Science Foundation long-term work that has been going on in the US. So some of these things that they monitor are pretty much common across the world. So that's why I sort of listed a few of them. One are cyclic changes. Cyclic changes in ecosystem and species, populations, communities. And one such thing that we do is of the phenology of the species. And then look at ecosystems lag. You know, there is always a history on any ecosystem, which is reflected only if you observe after a lag period. Like for instance, empty forests. We call about empty forests. You have forests, they're living, they're great, they look at it, but they're actually dying because there is nothing there to regenerate the forest. So vegetation largely fits into this kind of processes. And then looking at communities as such, concerning stability, structure, and the development of the system. And all now can be pocketed into conveniently into a resilience framework. And then look at what to monitor in terms of species. What species are sensitive to change, and what habitats are sensitive to change. And this becomes very largely used as indicator concept in the long-term monitoring. So a lot of these, most of these are common. Most of these have been done in A3, and that's why I took that kind of definition. And very briefly, I don't want to go into great detail about what's happening globally in India. There are several long-term programs globally, and there are none in India. So, except for now a lot of initiative to start a new one. I just looked at TEAM, which is the Tropical Ecology Assessment Monitoring Network. This has been going on for quite some time. Conservation International being responsible for that. And the long-term ecological work at the Smithsonian Institution, which is largely focused on the Barra Colorado Island in Panama. And that has resulted in what's called the Center for Tropical Forest Science network of large 1 hectare plots. Sorry, large 50 hectare plots. And in India, I will not go in great detail because Jagdish will be talking a little more about the Indian monitoring program. But one thing is very clear is monitoring of natural systems has not been comprehensive in India, and nor has been done under a long period. We have only 150 hectare plot, that's in Mudumalai. And mountain landscape, we have several fantastic mountains, Western Ghats, Eastern Ghats, Himalayas. There's no mountain monitoring happening there. And long-term monitoring of processes like phenology has never been done. And though there has been some efforts to start such thing more recently. Globally, now what has, one could ask, why do long-term monitoring? It's obvious for most people, but just to give some idea of what it has revealed, largely from a forest perspective, there could be a lot of work from hydrology, water, etc. But I'm looking mostly at the the forest part of it. Barra Island, most of you, or some of you probably will know. It's a very interesting small island in the Panama Canal, where the Smithsonian started its work almost 30 years back, and has been consistently monitoring that little patch of forest, and has revealed amazing amount of details about how tropical forests actually function. So, this is clearly shows that how the trees in the Barra Island, consistent trend towards increased drought tolerance and increased average food density. It's all possible because you've monitored them for several decades. Pathogens play an important role in keeping rare species rare. So, and then forest dynamic plots, this is what they call it, the 50-hectare plots, provide a detailed baseline for understanding long-term changes in forest carbon storage, information that will be critical for understanding impacts of global change. And species assemblage in forest dynamic plots are influenced significantly by species input from the surrounding community. Now, meta community. Now, this is some of you who have done community ecology will very clearly say this led to what is called as the neutral theory by Hubble, and that's all possible because they monitored these plots for several decades. And closer home, Sukumar and colleagues from CES found that evidence that current frequencies of fire and large mammal herbivory, largely by elephants, to convert the Mudumalai forest to savanna or grasslands. There is no evidence for that. So the popular conception is that elephants and fire could lead to a lot of changes in the thing, but long-term monitoring, almost over 20 years now, is slowly giving evidence that those may not be real factors. Okay, and then of course the more recent picture, thanks to Priyan who sent this to me, the monitoring of insect. When you talk about trees, you talk about elephants, you talk about tigers, but you don't talk about small things that run the world, as Wilson said. They found in Germany that the population of insects are going down, and then they came up with this fantastic, completely sensor-based monitoring network that they're planning to put in. Okay, so that's about what has been happening generally on the long-term monitoring side. Now I will go on to what we have been doing in this in A3 for the last so many years. Well, there is, I define this as a pre-A3 and post-A3. As you know, A3 started in 1996, and so we started monitoring in almost 6 years before that. But the one common thing is Professor Kamal Bawa here. Sorry. Why we started long-term monitoring? I mean, we never thought that's going to be long-term monitoring, because okay, we want to set up, we want to study ecology, we want to go to the rainforest, we want to study the plants, we want to study the birds, we want to study the animals. That was the objective. There is no other big great objective we had. But that was facilitated by Priya Davidar, who was my guide, supervisor at Pondicherry University, and who was equally fascinated by going into the forest and looking for trees, looking for birds, looking for animals, etc. like that. So that was a really great motivation. Then came Kamal Bawa with his team. I mean, my first impression of Kamal Bawa was very different, in the sense that we were sitting in a small shack. If you remember old A3 building, it was something like that, but it's not A3 building, it was Pondicherry University. That was in a small building. And in walks in a man in a red shirt, and just starts talking. So how can an ecologist be wearing red? You have to be somewhere merging with the environment or wherever you work in. So, and then he talked about flowers, he talked about canopy. I think some of that inspired some of my colleagues to plant trees. So those are the motivation factor. And then came Richard Primack, who was a postdoc of Kamal Bawa. And I'm talking specifically to KMTR there, because, and Richard came and he saw us stay in one of the small quarters in the electricity board, and it was a cold, because where we work is pretty humid and cold. And he said, Kamal, I want to see where your students live. He said, no, they're down there. No, let's go down there. So he came, he pushed, he just dragged Kamal Baba, let's go see. And he saw this absolutely barren, what do you call, the stone floor. And he said, no, you can't let your students sleep on this cold floor. Come on, provide them beds, provide them cots, provide them this. So that was a real thing, because then the shack became a little more cozy. Probably that helped us to stay there and do the monitoring. Peter Ashton, of course, from, he never came here, but yeah, he was very instrumental in BRT. He provided that impetus that's needed for all of us to keep going. So that was a little story of the pre-A3. We continued from there. And then, of course, post-A3, we have the Vemanad flagship kind of work in wetlands. And of course, there are other areas that are slowly coming up. Okay, so what did we monitor when we started in the early 90s? So the early 90s, globally, there was a lot of talk about forest dynamics. But in India, we were largely confined to enumerating trees, putting up small plots, putting up transects and say, okay, this forest has so many species, this forest has so many species, etc. This is the abundance of species now. But we never looked at how these species change, how their population change, how the community change. So that's what probably not inspired me or inspired the thing, but that's what made Kamal Bawa, Priya Davidar, and Roffali. By the way, many of you know Roffali. Roffali and Priya Davidar actually had this project on Agasti Malay, funded by the Ministry of Environment and Forest, and they wanted to study the ecology of the rainforest. So it was all part of that, and so we started setting up some plots to study forest dynamics. At the same time was tropical phenology, because a lot of this work, tropical phenology, happened at Kamal Bawa's site in La Selva and in BCI in Panama. So he came from there and said, oh, you have to start this. This is something that is going to be very important in the long run. And at the same time, Southeast Asia and Malaysia, there was a lot of effort to look at tree phenology, especially for dipterocarp forests. Apana and all those people who pioneered some of the work in the canopies also looked at long-term phenology of dipterocarps in forests. But then there was no study in non-dipterocarp forests about phenology anywhere in the world. So that sort of made us to think that no, we should mark these trees and look at their. Okay. So the broad question that time was not really that time that we thought of these questions, but this is what sort of evolved over time, is to look at anthropogenic factors, including forest disturbance, climate change, on phenological patterns. Now, when I talk about anthropogenic factors here, in the 1990s, we were talking about disturbance, because forests were just recovering from a lot of logging, and we want to see how that disturbance actually affects the phenological patterns. But the climate part actually came in much later. And the second thing was, how do forests change over time in terms of recruitment, mortality, and turnover rates of the trees? And more recently, how do invasive species and fire affect these dynamics? So at the same time that ecological monitoring was happening, BRT was actually looking at community-based monitoring initiatives. And this has been pretty unique, because Kamal Bawa, I remember he telling us, you know, this book from Neotropics. It talks about communities, how they can help in not only telling about the forest, but actually monitor these changes in the forest. So, well, in KMTR it was not possible because there were no communities living inside to do that. But BRT was an excellent example where he put in a lot of efforts, and I think Siddhapa here would talk a little more about it in his presentations. So that's what started in 1990s. It was a kind of an experiment to involve communities in monitoring, and it has it's gone on. They monitored, of course, AMLA. They monitored beehives using local communities, and now it has been scaled up to Hills and also Kaveri Wildlife Sanctuary. So this has been a very interesting stuff that's been going on. And then the wetland monitoring. This is all post A3. The the whole idea of monitoring wetlands was because the wetlands are close to people, so use people to actually monitor them. So that's the community-based monitoring that we've been involved both at Vemanad and KMTR. Vemanad, you all know the fish counts and the water quality count. The Jaladapanam is, I mean, you keep hearing it every year. The effort that's being done using local communities, panchayats, and all that. A similar work we do in KMTR in terms of the annual waterfall census. Madhivanan will be talking a little later, where not only kids but also other local communities help us in generate data. Well, it's easy to say to involve communities and generate data, but we also have to keep a check on the kind of data that's generated. So good quality data that should come out from community monitoring is what was our focus. And I think Madhivanan will tell a little more about how we sort of kept that quality check on the data coming in, because what we did was 80 people monitored along with the community, and then we compared our information and see and slowly how 80 wants to withdraw from such initiatives. Okay. Okay, more recent initiatives, of course, grassland, the Bani grasslands by Abhi and Ankila. And of course, I can't talk much about water. There's a lot of instrumentation done by Jagdish, Veena, Sharath, and Srini. But I will just leave it at that because that's probably for a different talk. Okay, so putting all this together, what does AT monitoring do is, of course, we focus on pretty much large taxa like trees, birds, fishes, bees, and etc. Major ecosystems, forests, wetlands, grasslands. And also the kind of monitoring we do is, in most places, is socio-ecological in a sense, except in KMTR, right inside KMTR, where it's largely ecological. So I come to the monitoring that we do in KMTR, Kalakkad Munnar Tiger Reserve. This is the Agastyamalai Mountains. This is the Agastyamalai Peak, which is not exactly inside KMTR, but it's outside in Kerala. What is unique about KMTR is its large continuous patch of pretty much undisturbed wet evergreen forest. Though there has been a lot of plantations inside, both private and government plantations, but these are relatively small compared to the larger landscape that's available here. So it was an excellent place for actually set up kind of monitoring in 1990s. I'm not sure I would do that today. So, okay, a little more history. Kamal Baba, of course, gave us a lot of impetus to do that. But I don't know how many of you have seen this book. The page of the cover of this book, Ecology of Tropical Forests, Seasonal Rhythms and Long-Term Changes. This is it was considered a very classical book on tropical ecology, written by Egbert Lake from Smithsonian Institute. A fascinating mathematical ecologist. I mean, somewhat in terms of Mahathir, but if you get a chance, there's a next version of this book as well. Just read this book. It gives you what tropical ecology, what people thought about tropical ecology about 30 years back. So this was an inspiration. Because Priya Davidar who just landed up from Smithsonian there, and she was just working in these plots and this phenology and she was so excited. She got a fresh faculty position in Pondicherry University, and she wanted to just set up another BCI in India. So we tried in Andamans, which didn't work out. So we thought, okay, Agastyamalai is another point where we start. And so much of it, what we did came from this book. And of course, we have, listen, sorry for that picture, colorful picture. And of course, three of us did our PhD and started our work in that rainforest. Okay, so what did we do in KMTR? One was looking at long-term phenology. As I said, look at factors, anthropogenic, basically have forest disturbance, how it affects the patterns of these trees, and what are the ecological processes behind these patterns. One of the problems with phenology is, I'll come to it a little later, is that people look at flowering, fruiting and all, but they don't actually relate to other processes that happens in the forest. So we monitored about 800 canopy, subcanopy and understory trees. Set it up in 1990. We still continue to do that. Similarly, Ganesh will talk a little, much more about it, is setting up these permanent plots in the wet evergreen forest, which I'm set up in 94, monitored every 5 years. And this was the first kind of permanent plots that were established in mid-elevation evergreen forest in the Western Ghats. The French Institute had a fair amount of plots in the low elevation evergreen forest, which is a very threatened forest ecosystem in the Western Ghats. Of course, Sukumar and others have it in the dry forest, and much later on into the Nilgiris in the wet forest. But at that moment, 1990s, this was one of the first plots. So these are just the broad factors we thought would be affecting recruitment, right from ecological process, disturbance, climate, the functional traits of the plants, and the phenology. These are all mutually interlinked, so those linkages are not being provided here. Okay, phenology now. How many of you know what is phenology? I assume that if you don't raise your hand, all of you know. So, okay, this is what people thought of phenology. I mean, this is something ever since humans settled and started doing agriculture, they kept track of plants, when they flower, when they fruit, because it was essential for their own existence. But this is what was quoted in Bible almost 3,000 years back, is that learn a lesson from the fig tree, and once the sap of its branches runs high and it begins to sprout leaves, you know that summer is near. So this is how they started relating the cyclic patterns in trees. And then Aboriginal have their own calendars, recognized between 5 to 10 seasons, each season defined by changes of flora and fauna, as well as the cycle, wind, rain, temperature. I'm sure there are lots of things from India. Even the Soligas have their own recognition of patterns and times when trees flower and fruit. And but systematic monitoring, which is very interesting, is that the Japanese started monitoring this cherry trees in their own backyards way back 1300. So this is considered as the most long-term monitoring data that is available. Okay, now, but all of this, whatever is defined, has been largely from a temperate background. People work in Japan, people work in Europe, people work in the US. So what's happened to tropical phenology? It's, I mean, tropical phenology in the early 90s, or maybe somewhere in early 80s, I would say, people looked at phenology from a very different perspective. They looked at it from a resource for primates. Because primate studies started way back, early 60s. And that's when they started looking at, when primates started using leaves, flowers, fruits, they also thought that it's time to look at how these resources change. So a lot of primate studies actually led to phenological studies. So this is what has happened in many sites across the world, right from Central America, Africa, Southeast Asia, and Australia. Australia, of course, don't have primate, but it's for a different reason. And then one of these studies from Gabon in Africa, Chapman and all, who have been documenting the primate behavior in that area, documented phenology for the last 20, 25 years. So very similar to what we have been doing in India. Well, there are no long-term studies, long-term phenological studies from the wet forest in India, as I've already told. And what is known about phenology? Well, very, very clearly, phenological patterns are most diverse and least understood in the tropics. Even today, I think it's still remains at the same point. And largely because we don't have so much information on tropical long-term phenology. And then there was a paper by Collett and Frankie, which said that it's expected that the robustness of current phenological patterns to high interannual and spatial variability means that most plant species will not be affected by phenological processes due to climate. There could be other factors that could be driving these the survival of these plants, not only because of climate. And these things could either be interactions, ecological processes that are happening in the forest, and largely because when plants shift the flowering or fruiting, it directly affects the pollinators and dispersers. So that way they would be suffering in terms of pollinations. So there is an indirect loop that could affect plant regeneration. And that's what it says. Ecological processes that are very key, they're very important. So that's likely to be most affected by these changes in climate. Okay, so what did we, as I told you, just to give a glimpse of the phenological data that we have, both from BRT and KMTR. BRT, we had about 100 species, 700 individuals, and monitored across 23 years. What did we monitor in these things? Flushing, that's just basically the kind of young leaves that come out, and then senescence, the dry leaves, the dry flowering, fruiting. And in KMTR, we also included another factor, which is called the herbivory, how much of the leaves eaten. So this huge amount of records, because you have 5 parameters recorded across 700, 800 trees, across 12 months, and across 20 years. So if you just do the multiplication, it's a huge amount of data. And so we also had climate. KMTR, we had the, fortunately, we had the estate there, which produced some very reliable rainfall and temperature information. Okay. So as I said, there's a huge data sets, and it takes a huge amount of effort to actually analyze and not only analyze, it probably doesn't take so much time to analyze the data, but just time to consolidate the data. So we're still in the process of consolidating data. So what I'm presenting here in the next couple of slides is a very broad, very coarse kind of results. So you can't really interpret it very deeply into it, but you can just give you a broad pattern of what's happening in these forests. One is looking at patterns. You can see there are differences in pattern. There are species that fruit annually. There are species that fruit once in 2 years or maybe 3 years. And then there are ones which fruit occasionally, once in several years or maybe even more than 10 years. So we divided that as something that's annually fruiting, as something that is supraannually. So these are really data that we have recently consolidated across what could be about 20 years of data. And this is only one subset of the data. We have a larger subset which is still to be consolidated. So there are differences in patterns, which is well known. The other thing, most of you would have seen this graph. This is the work of Subhadra in 1999. How bees, social bees, are very important for most of the trees in the KMTR, and more importantly, the social bees are migratory. They don't come, they're not found in the place all the time. And they come into it because of the flowering of one species, Palacium ellipticum. And once it starts flowering, the bees are there, and then there are several other species that benefit from its occurrence. So this is a pattern that is sort of repeating. So it is something that you don't require 10 years of data to actually tell this. Okay. Now there are other patterns. This is Calophyllum astroindicum, which is endemic species, also bee pollinated. It's a small bee pollinated, if I'm right. But it has a very supra-annual kind of flowering. So it had flowered sometime in quite a good flowering in 2007, but after that it has been not really been a very good flowering. Actually, this data is from 95. Actually, in 94 there was a pretty massive flowering that happened in California. So if you take 94, 2007, this is a huge difference. So Calophyllum is a bee pollinator, as I said. So how does it attract pollinators when it flowers once in once in 10 years, 20 years, or what is the thing? And also, I want to add here that Calophyllum is not a very common or dominant species. It's very sparse. You can see these are the flowering, one individual here, one individual there, and there's nothing else. So there are very small patches of these trees that flower. So what we assume, we still are not very clear, that's why I said the data is very coarse, is there are species that flower every year. And these species also attract bees. So whenever whenever Calophyllum flowers, there's always some subset of species in the community that flowers, and that those the bees that come into those flowers come also to Calophyllum. And what interesting happens is that because Calophyllum sometimes is mass flowers, there's a huge amount of increase in bees. So the colony size increases, the number of beehives increases, etc. Okay, so this is one thing, and the other thing is this is again something that all of you would have, most of you would have seen, is the episodic response of the frugivores to fruiting. Palacium ellipticum, I already mentioned, synchronizes. It's an annually flowering species. You can see this yellow line, which flowers almost every year. There are years which it doesn't flower, but then there are years when everything flowers. Every individual in a forest flowers. It's called the masting, and so this happens not very often. And when masting happens, we found that there's huge amounts of bats coming. We don't know where they come, but they are there. And then they eat the fruits. It's bat dispersed, species, and they disperse the seeds. So when I first presented this, so some of you outside, wow, this is so much. Or if you don't monitor, if you only do a monitoring up to here, you're going to miss out this. If you start from here, you're going to miss out that. So, I mean, it clearly says that long-term monitoring is very, very important. But this kind of cycle, have we seen it? This happened in 1994. We haven't really seen this repeat. Okay, I mean, 1994, now 20 years. We haven't seen it repeat. And now, was this just a random stuff that happened once in a while, or is there something else to it? So, in 2015, a similar thing happened. But what happened was that in 2015, we had lots of, almost all species, all individuals of Palacium flower, did fruit, and we had the bats coming in, and there was also a lot of dispersal. But then we also looked at some of the other sites. This is one of the sites. We looked at two other sites where a similar thing is happening, and we found that the intensity of flowering in these sites were not so high. So there was this patch asynchrony that's happening across this landscape. There are some patches which are synchronized in flower, and some populations flower, some populations are completely different patch do not flower. So what we assume or presume now is the bats are there in the forest, but they track these resources across years, and whenever there is a masting, there is a huge congregation of these bats and frugivores. The evidence not only comes from bats, we have a fair amount of circumstantial evidence. You know, long-term monitoring is you've been in the site for 20 years, you have some gut feelings. You have some, you're feeling of that forest, which is sometimes more important than what you actually measure by your instruments or by observations. So the same thing happened with birds, large frugivorous birds like pigeons. They also track these resources across space and across habitats as well. And these are completely different between years. So very similar to bats. So there is a small set of frugivores in that area, because Kalakad Tiger Reserve is, though it's very unique, very interesting and all that, we have seen both from a pollination point of view, from a dispersal point of view, there are only limited number of species and limited populations within the reserve, which has to serve this entire thing. So that's why we feel that these limited populations actually move across the reserve to, depending on the synchrony of the flowering and fruiting, to maximize the fitness of the plant as well as themselves. Okay. And then some random events. I don't know whether you want to call it random, but this is what happened. In 2016, Mystica dactyloides, which is related to the nutmeg, the popular nutmeg. Sorry, not dactyloides. It's the old name. Now it's called Bedomii. Was experienced mass defoliation, largely because of one little insect called the, I don't know how you pronounce that, Sastroidus visuati. So this happened in I think in April 2016, and by end of that year, the trees were completely defoliated. And interesting thing is that when we didn't know what the species, what the insect pest was, so we sent it to Pratapan, who is an entomologist specializing on these insects. Thanks to Priyan for that. And he found that he's the one who said this is the species, and he also said that this was recorded on the nutmeg, the exotic nutmeg that is grown in Kerala, and it is a major defoliator of this nutmeg. And this was recorded only in 2015, even on a nutmeg. And in next year, 2016, it's happening on a native species, Mystica bedomii, which was completely unknown. So Pratapan said, we don't know what is the host species, but now we have good evidence that there is a host species for this within the country. So these are something that we never expected. And the other one, which we know nothing about, is the spiny dormouse. The spiny dormouse is in the months of May and May and June, exclusively depends on Mystica fruits. It's if you want to see a dormouse, the best time to go to KMTR is in May. And you put a couple of these fruits and you watch with your torchlight, you will definitely see this mouse. Otherwise, it's extremely elusive. You know, me and Ganesh and Subhadra have tried for years to photograph this until we tracked this with the fruit. So it seems to be exclusively dependent on it. I mean, we also had captive dormouse. We gave them a lot of fruits, but it's just gorging on those fruits. So due to defoliation, there was no fruiting. There was absolute fruiting failure across the forest. And what happened to dormouse is not anybody's speculation. Okay, so finally, we'll touch on the phenology and climate. Garima will be talking a little more about this, how climate has changed in that place, especially rainfall. But what has come out is that there is increase in dry season rainfall, or rather the variability in the dry season rainfall has become higher. There are two dry seasons in India. One is March and the other is in August, September. So there is a slightly higher rainfall happening in these two periods. And what's the consequence of this? Very obviously, the flooding happens during dry season, and it's likely that more rain would affect flooding. And why I say that is because there is evidence from our work, which we have written up in the A320 book, is one of the dominant species, Cullinia axarellata, which flowers during the dry season. Very important resource for the frugivores. We did a water experiment on it. We actually sprayed water on the flowers, and we found that most flowers did develop fungal pathogens and did not actually fruit or did not attract frugivores. So there is some evidence that this could affect both the frugivores as well as the. Okay, I'll come to the last few things about. I'm actually running on. I have only 40 slides. So what it takes to do an LTM? Well, okay. Well, everybody will think that funding, but actually it's not funding. It's the commitment of an organization and individuals is the key. Because unless you're committed to doing that, you're not going to do that, no matter how much funding you will get. Your professor tells you to go up there and look at phenology. Oh yeah, let's go there and come back. But actually going there and actually doing it is very different from actually going and coming back. So there needs to be a commitment from organization and individuals. I know, I mean, the KMTR work, many times we never had any funding, but we had that motivation to go up and look at what's happening to the trees. Well, this is very obvious. No agency actually in India funds for long-term work. They want results in a year or two. So that's another thing. Permits are usually not given when you go and talk to first department. No, what do we get in 3 years, end of 3 years? What do we get in end of 2 years? No, this is not useful for us. So, especially when you come to phenology, you need a trained team, because if you have you can use a lot of volunteers to do plot work. I mean, because it's just measurement and stuff like that. And if you have a very good botanist to tell you what the tree is. But phenology actually is measurements which you do with your binoculars, with your eyes. And so that to remove any kind of individual bias, you need to have a particularly dedicated team of a few individuals. So that has been a challenge for us. The early years, most of us were doing it. And then we had to train people who kept doing it. So that bias is completely removed. Almost removed, not completely, but yeah, so we need a dedicated team. Data management is critical. We are realizing that we have a huge amount of data, what to do, how to analyze, what to put it. That's a huge pain. And this is something that we've been asked repeatedly, not only from A3, but even from outside, that what are you doing with your data? So there has to be a real effort to publish, because we always tend to say old data, okay, let's keep it, we'll do it later. So that kind of procrastination sets in. And it's, as I told you, it's a sense of attachment to a place. I mean, your attachment to a tree, attachment to an animal, things like that actually motivates you to do and monitor. And also critically, who listens to you? I mean, if you monitor tiger, everybody will put you on paper. But if you look at phenology, nobody's going to look at you. I mean, it's not very critical. So that's the thing. And what the way ahead? Well, I would I keep emphasizing this, because it's a self-driven motive to get rid of this data, get it out into the public. It's not only for KMTR, but I'm sure it's for BRT, MNR, and many other sites that will come up in the future. Continue monitoring, because people say, okay, you're 20 years monitored, let's stop. But actually, the whole 20 years of monitoring might collapse if you just stop. Because there are some patterns which you will not be able to see even in 20 years. Continue monitoring, but also include a little more of social, economic, biophysical parameters, including new taxa and also new sites. Landscape element has been missing from most of this monitoring that I mentioned. We have not looked at how the landscape around has changed. That we're doing it now. Well, there's no funding, so I think an institution should create an endowment for monitoring, because monitoring has been the source of because A3's flagship program, monitoring, so needs some support. Of course, linked to national and international efforts. Well, global phenology monitoring is one thing that I think of, but there are several other efforts that have been done across the globe to which we can link. We tried in the past, sometimes it fails, sometimes it works. I want to thank several, I probably have missed out all those people who have funded and helped us, but that's it, thank you. And our field station has also changed over the same period of time.
I'm not a scientist, so forgive me if the question sounds stupid. But you mentioned that you have been monitoring two kinds of anthropogenic changes. First, to do with more localized disturbances, and then later including climate change factors. Does your data show the relative importance of one versus the other? Are you able to track that?
No, we would want to track that, but you haven't done that. Some data that are not presented from some of this data that Ganesh has is when you say disturbance, say plantation disturbance, like what's been cleared and it's been planted. This will be done in the late 60s, stuff like that. So monitoring that, we have also been monitoring, Ganesh especially has been monitoring that change, whether species have recovered or the populations have recovered and all that. But even after about 40 years, of course, the species have come and all that, but it has still not completely reached what you would expect a fairly good so yeah, the small scale disturbance is still having a lot of effect. Well, we really don't know climates or really constant, what is the relative forces?
Hi. So thanks for this nice presentation. I know somewhere like we could adapt some 15, 20 years of satellite data to encrypt this your findings and some mathematical models to make it better. So how you are thinking about, is it possible to use remote sensing parameters to make the model or make the results better?
Well, that's a good question and interesting question as well. We've been pondering at that for some time because a lot of phenology is not done with, especially visible images, high resolution images, and all that. But maybe it's possible in the dry forest, But our pitch is that in a rainforest, the dense canopy, a lot of flowering happens sub canopy. A lot of fruiting happens in sub canopy. Whatever high resolution images you might have, you will not be able to capture what's hidden below the leaf unless you can look through the leaves. So that's, I think, is a quite an indication. I don't know if the remote sensing people will be able to help with that. But so that's, I mean, I feel is a limitation because we tried some kind of correlation between species that actually flush. I mean, like I showed you the flush, which can be easily be seen from our remote sensing imagery, and related with the flowering and all that. But it didn't work out. We didn't pick out most of the even the flush. Maybe with high resolution, that is possible. But actually looking at fruiting subcanopy trees, understory trees, for instance, it's not seen at all. It's not possible.
Any more questions?
Yeah, thank you for listening.
Thank you, sir. May I now request Karthik to give away AWS token to TG? Karthik? An AWS token to TG. Thank you, sir. We had always enjoyed your ecology classes. So we come to the end of the first inaugural talk. We will now break for high tea, which is between 10:25 and 11. Before we break, there's a couple of housekeeping announcements. Sorry, Karthik, did you have something? So the media team would like me to inform you to avoid this area. Please do not share space with the presenter. We would also encourage you to collect your lunch coupons during this time. The proceedings book, a soft copy of the proceedings book has been emailed to all of you if you would like to look it up. For the next session, we have session coordinators, and Anup is the next sessions coordinator. He would be Anup, if you can show yourself. So as you know that the talks are 20 minutes, which is 15 plus 5 minutes for discussion. At the end of the 13 minute, Anup will show you the presenter's sign that says 2 minutes left. And also during the discussions time, please adhere to time. It'll be great because we have a packed session ahead of us. So thank you all. See you back at 11.